This is Barker's neuron, a drawing of the archetypal neuron cell made by Llewellys Barker, a draftsman, and published in 1898. It shows an ideal of the neuron cell type. This is the more contemporary textbook image of a neuron cell. Barker's drawing is its parent, and it is still like that, an ideal or a set of criteria defining the characteristics of the neuron cell type. This image is more than 100 years old, but it is still relevant today, even if no active neuroscience researcher would expect a neuron cell to look like this. This is the Spanish scientist Santiago Ramón y Cajal. Cajal was the first person to see the neuron cell. In 1906, Cajal, along with Camille Golgi, who invented the staining method which Cajal used to make his observations of the embryonic brain, was awarded the Nobel Prize in recognition of work on the structure of the nervous system. Cajal saw what no one else could see, that the brain was composed of cells, neuron cells. In formulating this principle, Cajal effectively unified the science of cell biology and laid the foundations of modern neuroscience. Many people had inspected the tissues of the brain. Only Cajal was able to see and draw the cells which we now understand to be the units of its composition. These are just some of the images, the slides and the drawings which Cajal made, usually at his somewhat improvised laboratory or at his kitchen table, working long into the night after his children had gone to bed. Cajal's vision encompassed aspects of the growth, regulation and function of neuron cells. But it is only more recently that neuroscientists have begun to be able to visualize the life and animation of the neuron cell directly. This moving image was produced by Darren Williams and his co-researchers. Darren is just one of many hundreds, perhaps thousands of developmental neuroscientists at work today, who are attempting to add new data, new experimental realization to what Cajal set in motion so long ago. Darren designs flies. He uses genetic recombinant technologies in order to be able to visualize the process of neuron cell development, of growth, of regulation, of function. We might say Darren uses biology to study biology. This image was produced by one of Darren's colleagues. It is of the neural network of the fly Drosophila. The neuron cells and their connections have been painted with the colours of fluorescent protein derived from jellyfish.
Darren's experimental work is painstaking. It is meticulous, precise, often protracted. Nervous systems were dissected from larvae and fixed in 3.7% buffered formaldehyde for 1 hour at room temperature and then washed 3 times in phosphate buffered saline at pH 7.8 with 1% Triton X 100. Fixed samples were blocked in 2% normal donkey serum supplied by Jackson Immuno Research Laboratories, West Grove, PA, USA, in phosphate buffered saline for 30 minutes, and then incubated in primary antibodies for 1 to 2 days at 4 degrees centigrade. After washing out unbound primary antibodies, tissues were incubated overnight at 4 degrees centigrade in a 1 to 500 dilution of fluorescently labeled secondary antibodies. After repeated washes with phosphate buffered saline, tissues were mounted on polyxylene coated cover slips, dehydrated, cleared through xylene, and mounted in a xylene-based medium supplied by Flocker Bax of Switzerland. Individual fluorescently stained nervous systems were imaged at ×60 magnification using a Zeiss confocal microscope. Z-stacks were collected with optical sections at 1 micron intervals. In collecting the Z-stacks, the excitation wavelength was optimized for the respective fluorophore to avoid bleed-through. Raw data stacks were imported into NIH image for viewing. This result is just one tiny drop in the ocean of contemporary neuroscience data.
This, and much more research in developmental biology, is consequence of genetic manipulation, recombination, using biology to study biology, and use of the green fluorescent protein is a modern landmark in that. In fact, this technology itself is a Nobel Prize winner. Darren's research work, and the research which he shares with his students, is essentially imaginative. But it is also material. Darren and his students, his co-workers, seek to understand aspects of the neuron cell identity which are still unknown or hidden to science. The imagination that they use, the vision necessary to project what the neuron identity might be, is firmly rooted in experience of experimental craft, however. We might say that Darren's imagination is anchored in material plausibility. Darren's insights and his making of new data are coupled in the lecture theatre, in his laboratory and in his office. A new idea arises, sometimes ephemeral, sometimes leading directly to a new experiment. What is critical in Darren's work and in Darren's reading of the research work of other scientists is his being able to judge what is plausible and what is useful. Also, of course, what is not. In this way, Darren is looking for the neuron cell identity to show itself. And it is this search that he shares with his students and his colleagues.